Welcome. My name is Christy Trifone-Millhouse. I'm NISAW's Executive Director, and I'm so excited to welcome you to the 2024 National Invasive Species Awareness Week. Our theme for this year's event is Protecting North American Biodiversity. NISAW is an important international event powered by the North American Invasive Species Management Association, or better known as NISAW, to raise awareness about invasive species, the threat they pose, and what can be done to prevent their spread. NISAW provides a platform for individuals from local, state, federal, tribal agencies, regional organizations, private industry, and citizens to discuss legislation, policies, resources, and improvements that can be made with elected officials and decision makers. We have quite a week planned for NISAW, with presentations focusing on language and sharing best practices for communicating about biological invasions to scientists, stakeholders, and the public. We will learn about federal priorities for research and management. We will hear about risk assessments and how they help us understand species movement and potential in diverse communities. Wrapping up this week, we'll learn about on-the-ground efforts that are helping to enhance biodiversity and habitat quality. This is a week not only for learning, but for action. Throughout the week, we will be sharing links with you in an effort to connect you with your U.S. senators and House representatives. We want them to hear our collective voices, to learn about the importance of this work and why we need to support and fund all of this to continue to press forward. Before we begin today's webinar, I would like to share with you briefly more about NISAW. NISAW is deeply vested in our mission to empower the management of invasive species in North America. We are led by an 18-member board of leading experts and professionals with supporting committees, a strong NISAW team, and a membership that continues to grow in numbers by the day. Our core programming is designed to provide our community with the connections needed to learn about advancements in the field and to share lessons learned. We also provide a collective voice for our community through events like NISAW. Lastly, we aim to provide the tools such as professional development, international standards, campaigns, and the NISAW Certified Weed-Free Products Program to broaden our impact and to help further our mission. If you are not currently a member, I strongly encourage you to join us. A membership investment helps us to continue to do our critical work as a leading 501c3. There are certainly many options to consider. Please reach out if you have any questions. Okay, it's now time for the main event. I'd like now to introduce Dr. James English to provide an overview of the session. Dr. English is the Biosurveillance Network Coordinator for the U.S. Geological Survey. Thank you, Dr. James English.
Thanks, Christy. Well, it's great to be here. This is our fourth year in a row that USGS Ecosystems Mission Area, specifically the Biological Threats and Invasive Species Unit, has been bringing these talks together during the week of Invasive Species Week. This year, we're actually doing something a little different to stick with the theme of this year's NISAW, or National Invasive Species Awareness Week. We're bringing you some talks that are a combination of general knowledge or specific science knowledge, along with some research that we've been doing. And we have some of our best researchers giving talks on a wide variety of topics that involve the nexus of invasive species and biodiversity. We're going to hear a talk about native Hawaiian birds and mosquito. Then we'll hear about trachinid mussels, then invasive grasses out west, Burmese python in the Everglades, and lastly, managing common reeds and how that impacts biodiversity. And we'll end it up with a panel discussion. And I'm here with Patrick Kowalski and Paul Heimowitz, both from the Biological Threats unit that I work in here at USGS. So with that said, let me introduce our first speaker. This is Renee Bellinger, She's going to talk about some work going on with invasive mosquitoes and their impact on native Hawaiian birds. And Renee is from the Pacific Islands Ecosystems Research Center.
All right, so my name is Renee Bellinger, and I am a research geneticist at the United States Geological Survey Pacific Island Ecosystems Research Center, located on the island of Hawaii. During today's talk, I will describe a novel conservation tool that is currently being implemented to reduce the impacts of diseases spread by invasive mosquitoes. But first, I'll start with a big picture view. The Hawaiian Islands, here, formed de novo in the middle of the Pacific Ocean through volcanic processes and are among the most remote land masses upon the entire Earth. The distance of these islands from mainland or continental areas led to infrequent founding events. The plants and the animals that did make their way to these islands encountered a novel ecological environment which released selective constraints and allowed for rapid diversification and speciation. Perhaps one of the most famous adaptive radiations is that of the Hawaiian honeycreepers, a lineage of birds that originated from a founding event by an ancestral rose finch. These birds evolved a level of morphological diversity that is on par with that of the entire passerine group across the remainder of the world. However, although the birds of Hawaii are famous for their uniqueness, they are also notorious for species loss. Of the more than 50 species once present across the Hawaiian Islands, only 21 species remain. And over half of those remaining are listed as endangered or threatened species under the U.S. Endangered Species Act. The Hawaiian Islands are one of the few oceanic land masses without native mosquitoes. This here, the southern house mosquito, is the culprit and the major cause of these birds' decline. The southern house mosquito was the first of 6 mosquito species to become established in the islands, having arrived in 1826. This is a tropical mosquito that is associated with humans, and it rapidly spread throughout the islands in residential and agricultural areas, and ultimately into the lowland native forests. And it's a little bit different from some other mosquito species that are super tied to humans. These will actually go into the forest and they live out in the forest at very low abundant numbers, but are very seasonally or can be seasonally abundant. So the declines in native bird numbers were observed at the turn of the century, along with the first records of bird pox legions, as is shown in that middle picture. The continued population declines and species extinctions followed. The first detection of the avian malaria parasite was reported in 1940s, but as with avian pox, it had probably been here for decades earlier. By the time the first comprehensive studies of remaining Hawaiian forest birds that occurred in the 1960s and 1970s, most species had disappeared from the lowland forest and had become restricted to higher elevation areas, which are cold and where mosquitoes didn't reside. However, now, due to climate change and the warming temperatures that are being encountered in Hawaii, these mosquitoes are further expanding their range upslope into regions of habitat that were once a refugia for the birds. These 4 species shown here to the right in photos, the akakiki, akake'e on Kauai Island, and the kiwikiu and okohekohe on Maui Island, are of most pressing conservation concerns because of their rapid population declines. The estimated 2021 populations were very small, and at that time, the estimated time to extinction was believed to be within a year or 2. And shown here under species, the estimated 2021 population size, the akakiki was just 45 individuals. And at this point, pretty much all of those individuals have been pulled into captivity to try and initiate a conservation breeding program. The akake'e, there was around 638 estimated individuals, and for the kiwikiu, just over 100, and the okohekohe has more in the thousands. But the time to extinction for these other species is within the next decade, if not within the next year or 2. So to address this critical conservation issue, the Department of the Interior developed a multi-pronged strategic plan that encompasses a breadth of conservation approaches to try and prevent the extinction of Hawaiian forest birds. So the goal of the strategic plan is, as stated, prevent the extinction of Hawaiian forest birds through the implementation of conservation strategies, integration of Native Hawaiian biocultural knowledge and practices, and the development of new technologies. There are 5 objectives to this strategic plan. However, due to time constraints, today I will only be talking about Objective 1, which is to develop and deploy Wolbachia-based incompatible insect technique, IIT, to reduce mosquitoes that vector avian malaria. You may be wondering what Wolbachia is, and I'll get back to that in a moment. But first, I just want to give a shout out to the very large scale effort that is being coordinated through the Department of the Interior and the Birds Not Mosquitoes Coalition, which includes agencies within the Department of the Interior, the state of Hawaii, NGOs, and private enterprises. And so the efforts that I'm describing for getting Wolbachia out on the landscape for population suppression of mosquitoes is coordinated through this group. Now, back to Wolbachia. Wolbachia is an intracellular bacteria that is naturally carried by many insects and some nematodes. An estimated 50 to 60% of insects carry this type of bacteria. It's passed from mom to offspring, and it can control the reproductive capacity of its host, meaning that it can impact whether or not eggs hatch. It can also make, in some cases, it'll feminize the offspring, kind of like thereby driving itself into the population. So for mosquitoes, when a male and a female mate and they have compatible Wolbachia, it will go through its normal life stage, which is eggs, then larva, then pupa. But when you have incompatible Wolbachia, what happens is the eggs fail to develop. And so the female will lay eggs, but the larva doesn't develop, and then you have no pupae, and it basically crashes the population. And so you can make this happen in nature by taking out the Wolbachia from one mosquito strain and reinfecting it with a different Wolbachia strain. So that's that incompatible Wolbachia technique, or IIT. This is not a genetically modified organism, and it's not a genetic modification of any form. It's just Wolbachia from mosquito 1 into mosquito 2. And this natural way to control insect populations has been used all over the world to reduce mosquito populations that spread human disease, but this technique has never been applied in a conservation context. And so, coordinated through the Birds Not Mosquitoes Coalition, we have for the past 7 years worked towards deploying this technique in critical forest bird habitats. Now, the upshot here is that by repeat inundative releases of mosquitoes on the landscape, the mosquito population sizes will crash. First, however, you have to develop the transinfected Wolbachia line, that's like step 1, in the lab, which has been done independently in multiple laboratories. We're working, currently the contract is with Verily Life Sciences, and they are mass rearing mosquitoes in basically a mosquito factory in California. The mosquitoes get shipped over to Hawaii. They're packaged for distribution in these little pods, and then we distribute them on the landscape. And by we, the work that is currently being conducted is through the Maui Forest Bird Recovery Project, and they are doing the on-ground implementation of this technique. So coming back to the idea of inundative releases, you have to be able to release the mosquitoes all across the landscape. And so here, these swath lines show little dots and dot coordinates, and you can see that some are brown and some are kind of a greenish color. We use an alternating offset approach to saturate the area, and so the brown is on day like on a Wednesday, and then that kind of greenish is going to be the drop points later in the week, like on a Friday. And so the mosquitoes get dropped twice per week in that area, and that's about a 1,600 acre plot. Now, this is what the actual landscape looks like. You can see it's extremely rugged terrain. So you may be wondering how we get these mosquitoes on the landscape. And the answer is, right now, helicopters. So you can see the gun here. That's that little arrow that I just popped up. And it's like a little air gun, basically, and you shoot these little mosquito pods out, and they're in a biodegradable package, and they land on the landscape. And that is how we are getting them out. As a part of USGS research, we've been doing baseline monitoring for seasonality and abundance of mosquitoes in Kipahulu Valley at Haleakala National Park on Maui for the past, well, we did a whole year of monitoring last year. That work was led by Dennis LaPointe and Catherine McClure. And then there was other research conducted in 2019. And so in this area, this provides critical habitat for 6 Hawaiian honey creepers, including the Akohekohe and the Kivikio. Recent surveys found that the population sizes of these species have declined more than 50% during the last 10 years. And so we are targeting the Kipahulu Valley in particular for this IIT effort. We've also been developing and testing ways to better trap mosquitoes, to measure abundance, and to characterize the success of the IIT program. We're monitoring for larval mosquito presence, and we're conducting eDNA sampling at USGS, and we're developing field protocols to estimate egg raft viability, which is a component of measuring whether or not the IIT approach is working. And then we're also doing mosquito abundance and avian malaria disease monitoring. And the adult mosquitoes that are captured in these areas are subjected to genotyping so that we can examine population genetics of mosquitoes. And so one of the projects that I'm leading is for mosquito genomics. I have, in partnership with the Kauai Forest Bird Recovery Project, Maui Forest Bird Recovery Project, University of Hawaii at Manoa, and U.S. Fish and Wildlife Service, I sampled 37 populations of mosquitoes, and we've genotyped around 1,000 specimens, and this analysis and modeling is ongoing. And we can use that information to try and understand mosquito connectivity and population sizes. So I want to end with the point that there are a lot of endangered species in Hawaii, but any mosquito abatement efforts that we conduct are going to help all of these different species. And so part of all of this effort is being funded through BILL, and these funds have potential to be transformative for our ability to prevent the extinction of Hawaiian forest birds. There are 2 species of Hawaiian honeycreeper that exhibit some tolerance to avian malaria, and their genomes and transcriptomes may hold the key for understanding how to develop tools to improve disease resistance. And so I didn't have enough time to go into all the details of the types of ways that the USGS is supporting these IIT efforts, but please do reach out to me if you have any further questions. I want to end this with acknowledgements, funding, the Bipartisan Infrastructure Law, Fish and Wildlife Service Science Support Partnership, USGS Biothreats and Invasive Species Program, Pacific Island Climate Adaptation Science Center, National Park Service Island Network Inventory and Monitoring Program. And these are my colleagues who are at USGS, Catherine McClure, Dr. Atkinson, Dr. LaPointe, Evan Paxton, and Karina Penzari. Thank you very much for all of your time. And with this, I will answer any questions. And there's my contact information should you have any questions.
Renee, that's incredible. What a great talk. All right, we got one question, one comment. Question, at least the first one here. Is there any concern for Wolbachia mutating once it's in the mosquito population?
Actually, Wolbachia is already present in mosquitoes naturally. And so the Wolbachia that we use for the transinfection line is from Aedes albopictus. And Aedes albopictus is actually already on the landscape in Hawaii. So we're not introducing anything new. There's not really any concern for it mutating. You know, these do mutate over evolutionary timescales, but it's not a concern. And there's no genetic modification whatsoever that goes into this process.
Fantastic. Thank you for that. There's kind of a comment. Maybe you'll want to respond to it. Light pollution at night doubles the amount of mosquitoes.
There's lots of different species of mosquitoes, and some are more active in the day and some are more active at night. So Culex it's actually known as a bird biting mosquito, and it's a nocturnal.
Awesome. Any other questions? You see the one there.
Oh, traditional integrated mosquito abatement measures being considered. Yes, actually there are. So BTI is a naturally occurring bacteria, and there is going to be a bacterial aerial spray treatment on Kauai using booms and helicopters. So that's kind of, I would maybe call that more traditional. Other than that, like dump out your water, don't let your animal troughs sit there, dump out the flower pots. Those are kind of also traditional ways. I'll go ahead and put my name in the chat. Please do reach out should you have further questions. Thank you.
Thanks, Renee. Fantastic. What a great start. All right. Next up is Diane Waller. Diane is going to talk to us about dracenid mussels, and she is coming from the Upper Midwest Environmental Science Center.
So I'm with the Upper Midwest Environmental Sciences Center in La Crosse, Wisconsin, and do research on both invasive and native mussels. So I appreciate the opportunity to talk to you today about both of those, and in particular, the effects that invasive mussels have had on native mussels. So zebra and quagga mussels, both in the genus Dracena, so I'll refer to them as dracenids, are indigenous to Eastern Europe and presumably came to North America in the ballast water of ships arriving to the Great Lakes. The first reports of zebra mussels were in the western basin of Lake Erie in the mid-1980s. Quagga mussels were then identified in 1991 in Lake Ontario, but they very likely arrived before that. These mussels then quickly spread across the US and into Canada. You can see the brown areas on the maps there for zebra mussels on top and quagga at the bottom. So far, prevention efforts have slowed their presence in the Pacific Northwest, so our fingers are crossed there. So dracenids have the qualities of many invasive species. They're sexually mature within 1 to 2 years and then have a very high reproductive output. A female can produce over 1 million eggs per spawning event. Their dispersal is aided by a free-floating or planktonic larval stage that's called the Bellinger. The Bellinger then settles on a substrate to begin a benthic juvenile stage. It secretes byssal threads to stay attached, and this leads to a lot of biofouling in the juvenile and adult stages. They're highly efficient filter feeders, removing algae, suspended organic material, and even small zooplankton. So these characteristics, as you'll see, give dracenids a major upper hand in competing with native mussels as well as other filter feeders. We do consider freshwater mussels, both dracenids and native mussels, to be ecosystem engineers. By that, I mean that they drive habitat construction and control the availability of resources to other organisms. Because dracenids can reach such high densities, they can completely re-engineer a system. The filtration rate of dracenids can reduce phytoplankton biomass by 1.5 to 4 times normal levels in a water body. So even though they're on the bottom and they impact the benthic zone, they also have a large direct effect on the planktonic community and pelagic zone. So as they're filtering large volumes of water, material moves from the water column to the benthos. This is known as benthic pelagic coupling. So many native filter feeders are outcompeted by dracenids and then generally decrease in abundance, whereas animals that are deposit feeders in the benthic area can actually increase in abundance. So they have caused a significant decrease in the diversity and abundance of zooplankton communities. Key ones in the Great Lakes that have been impacted include the amphipod diarrhea, which is an important food for fish, and the freshwater shrimp mysis. Also, as a result of this filter feeding, they can increase water transparency 1.5 to 2 times, which leads to growth of macrophyte beds and, in some cases, nuisance algae such as Cladophora. Dracenids selectively reject particles of a certain size, which includes some cyanobacteria. So they've also been associated with an increase in harmful algal blooms. So zebra and quagga mussels have changed the availability of food at every level of the food web. The graph at the top center here shows the abundance of prey fish in Georgia Bay of Lake Huron, beginning in 1976 to 2016. The arrival of dracenids is in the 1990s, so about midway on that graph where you start to see a decline in prey fish diversity, and abundance. So this has created a food web imbalance where there's less food energy in the system for predators, and it's had a domino effect up the food chain, particularly to some commercially important species such as lake whitefish and walleye. That loss of a food base also leads to decreased growth of these fish and displacement of them to other areas in a water body to find food. In addition to shifts in food resources, dry synods can physically degrade habitat for native species, such as spawning areas for lake trout and lake whitefish. Ironically, the non-native round goby, also an invader in the Great Lakes, has become a major predator of dry synods. Quagga mussels now make up a significant part of their diet. But the most obvious detrimental effect of dry synod mussels has been to native unionid mussels. Dry synods seem to be drawn to the native mussels over any other substrate. When they find them, they attach often near the siphons where a current of water is carrying food meant for the native mussel. There's been a number of studies that have looked at the metabolic costs of dry synod fouling and found reduced fitness and reproductive success of native mussels when they become infested at a high degree. Even when they're not attached, dry synods outcompete native mussels for food and space. So before the arrival of dry synod mussels, native mussels were experiencing major losses of biodiversity due to a number of stressors. Currently, across the globe, only about 24% of native species are considered stable. North America has the premier mussel fauna in the world with almost 300 species. Currently, the Fish and Wildlife Service lists about 106 of those as threatened or endangered, and that number is growing. They're experiencing chronic declines and mass mortality events, really in high-value water bodies for still unknown reasons. So in contrast to dry synods, unionids have a very complex life cycle that makes them extra vulnerable and slow to recover from a stressor. They're long-lived, some live 50 or more years. They're very slow to mature. Some do not begin reproducing until 10 years of age. And they have a parasitic larvae that's dependent on a certain fish host. A native mussel is lucky to produce one surviving offspring in a year, or sometimes even in its lifetime, compared to thousands of juveniles produced from one zebra mussel. So now I'm going to pivot to highlight some of the work that we're doing to try and mitigate the impacts of dry synods. Our broad goal for invasive mussel control research is to develop tools for eradicating new populations, also reducing established populations, and preventing biofouling in both closed and open systems. So no one tool is going to work in all these situations, so we're taking a multi-pronged approach. Our research is looking at more efficient but still selective treatments for dry synods. Some of these projects I'm going to mention are aimed specifically at protection of native mussels, but we also consider how open water treatments could be refined so that we reduce negative effects to native mussels and other non-targets. So tools can also be more selective by targeting delivery to mussels. Copper molluscicides are one of the most commonly used tools at this time for controlling mussels in both open and closed systems. The EPA registration allows the application of these products at up to 1 milligram per liter as copper. And at that level, it's quite toxic to many, if not most, organisms. So we've been working with the University of Minnesota's Aquatic Invasive Research Center and focused on reducing the amount of copper needed to suppress particularly established populations of zebra mussels. We're targeting the Bellinger stage with copper in the range of 50 to 85 parts per billion. So compare that to the 1 part per million that the label allows. The graph here on the left shows the zebra mussel settlement in a control, the blue bar, and a treatment bay where we use the low dose copper. In red, so in the year of treatment, we had no settlement, and then 1 year later, much reduced settlement with the population starting to rebound 2 years later. So we found these low doses did knock down recruitment for over a year while having minimal non-target effects. But it does look like retreatment would be needed 2 years later. Another way to potentially use less copper is to tailor the treatment to the water body. The availability and toxicity of copper can change significantly with pH, temperature, and cation levels. So we're using the biotic ligand model and multiple linear regression models to predict site-specific copper concentrations and reduce non-target mortality when treating a specific lake. The third piece of refining treatments was to determine an optimal time to apply copper. Our low-dose treatments unexpectedly killed about 50% of adult mussels in our lake treatments. So we wondered if this was an effect of their reproductive condition, and if so, could we maximize mortality at a certain stage in that reproduction? So we conducted a series of 7 copper toxicity trials from June through September. The graph on the right shows the lethal concentration that killed 90% in the purple line, and then 50% of mussels in the gold line, with the water temperature showed as the dotted red line. So the take-home message is that the mussels were most sensitive to copper when water temperature peaked. This also corresponded to peak Bellinger density. So managers may be able to just monitor water temperature to optimize the effects of a treatment. Another tool we've been working with is carbon dioxide. We began working with it when it was being tested as a deterrent for Asian carp, and found it was about 3 times more toxic to dry-cinned mussels than it is to native mussels. It is currently registered as a pesticide. We're still awaiting expansion of the label to other invasive species, but in the meantime, we are exploring applications of it for dry-cinneds. So one of those is injecting carbon dioxide under a tarp or mat. Benthic mats have been used to kill invasive mussels, but it does take weeks, and along with it kills other organisms. So the aim with the CO2 tarp is to place it over high-value habitats, infuse the CO2 for 4 to 5 days to kill the invasive mussels and not cause native mussel mortality. With funding from the National Park Service, we're exploring CO2 systems for preventing dry-cinned settlement in native mussel propagation areas. So many facilities, the Fish and Wildlife Service is propagating some endangered species of native mussels. So we're looking at use of a bubble curtain that could be placed upstream or around propagation areas to prevent settlement. This bubble curtain could actually be used more widely around any type of infrastructure. Another application of carbon dioxide unrelated to native mussels is in raw water lines to prevent biofouling, environmentally safer alternative to chlorine. So we've been working with the Bureau of Reclamation to develop inline systems for treating raw water lines at their hydropower facilities. And lastly, I'll mention research that is aimed at restoring an aspect of fisheries in the Great Lakes. This is a multi-agency, multi-year project that's led by USGS centers here in La Crosse and the Great Lakes Science Center, and is funded by Great Lakes Restoration Initiative funds. This is an experimental mussel control project targeting mussel removal at select lake whitefish spawning reefs, one in Lake Michigan and a series of reefs in Lake Huron. So in the next 2 years, we'll be pilot testing some potential tools, including large-scale systems for physical removal, benthic mats alone and with a control agent delivered under it. And we're also working with collaborators to develop new formulations of molluscicides that could be delivered to the bottom and then slowly release the toxin in the benthic zone. So we're really excited about this effort to try and restore an important ecosystem function to these areas in the Great Lakes, and expect to gain a lot of knowledge for application to other systems. So with that, I thank you for your time and want to acknowledge our funders, GS, GLRI, Park Service, Reclamation, and many, many collaborators.
Diane, thank you so much. Lots of applause for this talk. Really important work. Well, that was a great talk. Thank you so much. Got a question for you. Are there any
Okay.
concerns regarding adding CO2 to the water that might increase the acidity of the water?
Yes, it very definitely does decrease the pH of the water. The amount of CO2 that we're using, because it creates carbonic acid, is still a relatively weak acid. And for the amount of time that we would be applying it, it's either at a low level enough that the pH would still be maybe slightly below 7, or only down to around 6.1 for a brief period of time. And the CO2 off gases quite readily from a system. So that's one huge advantage, is it's not persistent in the environment.
Got it. Also, maybe you can drop your email in the chat when you get off in case folks want to contact you directly. Here's another question came in. How might these methods translate to uninvaded watersheds like the Columbia, should mussels establish there?
So yeah, a large system. I think this is one of the things that we'll be learning from this experimental project in the Great Lakes, is how well some of these control tools will scale up to use in much larger systems.
And even in moving systems.
Moving, exactly, yes. So the treatment in the Snake River, we're watching with copper. Natrix was the product, but it was a copper product. So we're watching to see what some of the long-term response of the system is to that as well.
Okay, great. Another question, similar, same thing for the Mississippi.
Yeah, the Mississippi, again, these are tools that are at this point very site specific, very limited in their application. And so in the background, there's development of other tools that could be more widely applied, like RNAi tools. And this is where we're hoping to go with some of the new formulations of existing molluscicides that could be more broadly applied over a larger spatial area to be effective.
Thank you.
Okay.
What a great talk. Thank you so much.
Thank you.
Just in case you came in late, let me say welcome. This is our fourth time. This is the fourth Invasive Species Awareness Week series of talks by USGS. And I'm James English. I'm the biosurveillance coordinator for USGS in a group called Biological Threats and Invasive Species. And I'm here with some of my colleagues. Patrick Kowalski and Paul Heimowitz are both online. Ask questions in the chat if you'd like, and somebody much smarter than me will answer them. But we're going to go on to the next talk now, and this is going to be Seth Munson from Southwest Biological Science Center, and Seth is going to be talking about fire-prone invasive grasses.
All right. Hey, everyone. My name is Seth Munson. I'm at the USGS Southwest Biological Science Center. And today I'm going to be talking to you about invasive grasses. And these are a global threat because they can reduce native plant diversity, alter ecosystem function and habitat. And as you'll be hearing a lot about today, they can increase wildfire frequency and intensity. So I'm going to hone in on the southwestern United States, but I will say that these different invasive grasses are a problem for other regions as well. The Lahaina fire recently was carried by invasive grasses in Hawaii. There's fires really all over the world that can be caused by invasive grasses. But I am going to focus on the southwestern United States, which contains a very large concentration of federally managed lands where some of you work or recreate. And so while these iconic landscapes contain some of our most treasured natural and cultural resources, many ecosystems are being transformed by invasive grasses. And there's many factors contributing to their spread in the Southwest. But one of the most important factors is the fact that the Southwest, as we know, has experienced rapid human population growth. And with this growth comes increasing land use disturbance. When we have more disturbance on the landscape, invasive grasses can thrive because they can rapidly exploit available resources and spread quite easily. Another factor is the Southwest is becoming more arid. So there's stress and mortality of native vegetation, which can leave room for opportunistic invasive species that can respond quickly if this ongoing aridification is interspersed with a wet period. So let's introduce you to the cast of actors in the Southwest. In the western regions of our most arid parts of the Southwest, we have predominantly a cool season or winter precipitation regime. And in response to this, we have a lot of winter or cool season annuals. Many of you have heard of cheatgrass, Bromus tectorum. There's also red brome, Mediterranean grass. There's others, but these are some of the main players. As we move east into the Sonoran and Chihuahuan deserts, we start to get monsoon rain or summer moisture. So our invasive grasses in these ecosystems are dominated by summer perennials, including various love grasses such as layman lovegrass and bufflegrass. So I think while these invasive grasses are problematic for a number of reasons, one of the largest ways they influence ecosystem and human health is their role in accelerating fire cycles. So as you can see here in the lower left, we have the 2020 Bighorn fire that occurred just outside of Tucson. The lower elevations of this fire had quite a bit of bufflegrass that fueled the fire. In the lower right, we have the Marshall Fire outside of Boulder, Colorado. This was a fire that there were a whole host of reasons why it was so catastrophic. But one of those reasons was the presence of invasive grasses that kind of created that fuel continuity in combination with really incredibly high wind speeds, just caused this fire to blow up and cause some losses of infrastructure and loss of life. So what causes fire to kind of creep into these typically more arid systems in the Southwest? So typically we have native vegetation. If you've spent time in the Sonoran or Mojave deserts, you'll know that the native vegetation is sparsely distributed. Most of the vegetation hasn't experienced a lot of historical fire, and as a result, these plants aren't well adapted to fire. However, we can get invasion by non-native grasses, and what this does is that the normally bare soil in between vegetation is now covered in these invasive grasses, which creates fuel continuity. So now that fires can move into the system, they spread rapidly with invasive grasses, and this can also kill our native vegetation that is not well adapted to handle fire. After the fire, these invasive grasses can recover more rapidly than native species and then invade into areas, causing a fire cycle in which they burn and then rapidly regenerate. And then in a matter of years to decades, a system that was relatively poorly adapted to fire is now converted to a flammable grassland. So you can see this can be pretty devastating to some of our arid ecosystems in the Southwest. So if we look at invasive grass abundance and the risk of their contribution to wildfire through time, we can see initial low abundance followed by this rapid exponential growth and then kind of a leveling off. And with this, our risk of fire increases, but also our control costs increase. So we're better off rapidly detecting invasive species when they're in low abundance before they spread so we can potentially eradicate them. So USGS science informs really all parts of these stages of invader increases in abundance. We work on early detection, you'll see in my next few slides. We monitor spread and evaluate treatment effectiveness. And then if we're at the high stage of abundance, we think a lot about wildfire risk and post-fire restoration. So let's start with detection. It costs a lot to send people out surveying for these invasive species. Oftentimes they start in really hard to reach areas. So we're using the power of remote sensing with satellites and different drone technologies to enhance our ability to detect them early. One of the interesting things about a lot of our invasive grasses is that they start greening up earlier. So the solid line here is a green up curve through time. And what you can see is cheatgrass greens up really early in the growing season, whereas our natives haven't gotten as green. And so we can use that unique phenological signal to detect cheatgrass on the landscape. And so Tara Bishop, a postdoc with us at USGS, developed different forms of detection across national parks on the Colorado Plateau. And what you're seeing here in areas of red are places where cheatgrass has repeatedly showed up in multiple years. And our park managers are using these maps to prioritize efforts to control the spread of cheatgrass in the park. On the right is a recent effort where we kind of use similar methods to detect red brome and Mediterranean grass in the Mojave Desert. And again, these maps are being used to kind of prioritize efforts. But importantly, these previous efforts have been at a fairly coarse scale. And if you look in the lower left picture here, you can see that sometimes these invasive grasses are really in quite small patches. So we need to have really high resolution to be able to detect them on the landscape. And so in a partnership with Yellowstone National Park, what we're doing is we're taking our detection algorithms here on the far right, and we're using higher resolution imagery so that we can detect these really small patches on the landscape. And this is obviously important because if you're in Yellowstone National Park, you have a lot of wildlife species that use these open grasslands for grazing, including American bison. So if you have, this invasive grass getting out of control, causing fire, you can diminish the forage quality and the habitat for a lot of these grass species. Another tool that we've recently started using is terrestrial laser scanning. For those of you who aren't familiar with terrestrial laser scanning, it's an instrument that emits laser pulses and it spins in 360 degrees. So when vegetation or another object intercepts these points, the terrestrial laser scanner produces a 3D point cloud of the objects that intercept it. And so what you're seeing here in the green is a scan that we did in an area invaded by lame and lovegrass. And so in the background, you can see that there's that connectivity, right? The invasive grass has gotten into the interspaces between our native vegetation and is creating quite a bit of surface fuel. In the foreground, you can see that the lame and lovegrass is more patchy. And so really we need this kind of high resolution depiction of fuels because oftentimes in these arid systems, what either causes a fire to spread or slow down is the presence of this bare soil, this bare ground. And so being able to detect it at really high resolution helps us model wildfire And so we can actually simulate fire spread across the landscape and understand areas that are at heightened risk. So here you're seeing a 2D model, but we're actually starting to do this in 3 dimensions. So we can incorporate important factors like the interaction between the land and the atmosphere during fires, and hopefully generate better predictive models so that resource managers understand where wildfire exposure is the greatest. Another thing that we're doing at USGS is trying to understand fuel or invasive treatment effectiveness. So our partners at Saguaro National Park have been implementing control measures for buffalo grass going back to the mid-1990s. Importantly, we just compiled all the treatment data that they've amassed over the last few decades, and we're trying to understand when and where their treatments have been effective. So in the figures off to the right, you can see we're in 2 different districts of Saguaro National Park. In the Rincon Mountain District, you can see that the chemical treatments have been more effective than mechanical treatments, whereas in the Tucson Mountain District, they both have been equally effective. So I won't go into the reasons why, but we're importantly figuring out what landscape attributes associated with these different regions of the park help explain why treatments are more effective in one place than they are the other. In a different effort, after the Kolob fire ripped through Zion National Park, fueled by cheatgrass, we worked with the park to understand whether or not the herbicide and seeding applications they were using were effective. So you can see here that, again, in 2 different parts of the park, we had varying degrees of success. And generally speaking, you had about a 1 to 2 window before cheatgrass could regenerate. So we're really trying to understand not only what treatments work, but also how long they're effective. But finally, if we're going to get to the stage where fire is a part of the ecosystem now, and in places where it typically hasn't been, what can we do about this kind of situation where invasive grasses are prominent? We have fire that can rip through the landscape. We really need to be able to bring back native vegetation into this kind of situation. And so at the Southwest Biological Science Center, we formed the Restoration Assessment and Monitoring Program for the Southwest, or the RAMPS program, where what we do is to strengthen restoration outcomes in the Southwest by providing science and guidance on effective strategies to help recover post-fire and other disturbance on the landscape. So we do a number of different activities, including synthesize the effects of historical treatments. We also think a lot about developing protocols to help our partners implement successful restoration strategies. But one of the activities that has picked up a lot of steam is our RestoreNet project. And so RestoreNet is a co-produced research network between scientists and resource managers that systematically tests restoration treatments and native seed sources across environmental gradients in the southwestern United States. So typically we tend to do restoration in a bubble, but this importantly is helping us expand to think across the entire Southwest and understand where treatments are effective on the landscape. And so I think really this has proven to be a success because we can iteratively test things and have our resource manager partners provide important feedback so that we can develop strategies to help combat invasive grasses and other disturbances. And with that, I will thank you for your time. And if there is time for questions, I'll take questions.
Seth, thank you so much. What a great talk and important work you're doing out there. Really appreciate it. Thanks for the talk. A couple of questions. One is, are there any plans to project shifts in invasive grass distribution due to climate change and the use of detection techniques for EDRR in areas where the grass is not yet present, but it's likely to shift?
Yeah, that's a great question. That is a small part of my research, but the Fort Collins Science Center has a really cool tool you should check out. It's called the Inhabit tool, and it uses data from resource managers and develops, using species distribution models, a kind of a area where invasive species can occur. And I believe Catherine Jarnovich is incorporating climate change scenarios and how invasives may move into new regions in the future. Good question.
Thank you. Another one. Can you talk about any partnerships you have with Mexico down in the Southwest?
Yeah, that's a good question. We do work a little bit with our Mexican partners on some of our borderlands research that involves thinking about fire risk. And certainly there's different land use that goes on down there. And we have thought a little bit about the invasive grass question, but we've mostly been focused on thinking about restoration strategies and co-developing ways to mitigate fire risk. But we would love to have more cross-boundary efforts in the future.
Thank you so much, Seth. Thank you for being with us. In our fourth time to do this collaboration with NISAW for giving a talk during the National Invasive Species Awareness Week. And this year, we're concentrating on invasive species and biodiversity and ways those two elements interact. And so we really appreciate you being here. The next speaker just really demonstrates how much everything we do is a collaboration. None of these projects are USG specific. They all have lots of partners on the ground with other scientists, with other stakeholders. And this next project is coming up talking about Burmese pythons and will be co-presented by Amy Yackel Adams and Kristen Hart.
Thank you, everybody, for this amazing opportunity to share our collaborative research with you. Kristen and I work at two different science centers, and we also work with an amazing team of researchers and biologists. And we're studying invasive pythons to inform management strategies that will ultimately help protect and preserve the Everglades biodiversity. So let's start with taking in this set of slides. These photographs depict the beauty, the vastness, and the diversity of plants and animals in the Everglades ecosystem in South Florida. The Greater Everglades are identified by the USGS as a priority landscape. Here, USGS works collaboratively with partners to provide the science needed for stewardship and restoration of this ecosystem. The Everglades represent the largest designated wilderness in the eastern US. It's the most significant breeding ground for tropical wading birds in North America. It's home to the largest mangrove ecosystem in the Western Hemisphere. It's the largest and contiguous stand of sawgrass prairie in North America. It's the water recharge area for all of South Florida. And Everglades National Park in southern Florida is listed as a World Heritage Site, a biosphere reserve, and a wetland of significant international merit. So the Everglades is a hotspot for biodiversity. It's internationally known for its extraordinary wildlife and high plant diversity. And I want you to just think of the ecosystem where you live or work, and then compare it to these numbers. This is a pretty amazing area with more than 360 bird species that can be found in Everglades National Park alone. There are more than 750 native seed-bearing plants in the park, and currently it has 68 species of federally threatened or endangered animals. So in this slide, you're getting a good look at one of the invasive species that is causing major declines in biodiversity in the Everglades. It's the Burmese python. However, we rarely get to see this invasive snake this clearly. And that is part of how they became such a problem in the Everglades. This is an extremely cryptic snake, and each of these photographs has a python in it. And you may be thinking they would be easier to find if you were physically standing there in the field, but it is not the case. You literally could be standing on a python that's almost 20 feet long and never have any idea. I can attest to this because I have done this as I've radio tracked this species. So let's focus on the photograph to the right. I'm going to give you guys a few seconds to scan for the Burmese python before highlighting its location. This low detectability coupled with our inability to trap this species makes for a very difficult management issue. So let's cover some of the basic background information on this invasive snake. So pictured here to the right is a time progression. So you can see the bright yellow ellipse down in the south part of Florida, and then what you see as the colors change to kind of that purple at the very northern edge, you can see the spread of this species over time. Back in 1995, 2 Burmese pythons were located in Everglades National Park. And as I mentioned just a few seconds ago, that's highlighted by that bright yellow ellipse. The initial sources are thought to be pets released in Everglades in the mangrove swamp near Flamingo Point. And that was thought to be prior to 1985. This species was historically traded for a skin. Then it became a hot item in the pet trade. Because of the snake's crypsis and nighttime activity, its population continued to grow and expand. Between 1995 and 2022, they've been observed every year, and this includes all the colored zone on the map to the right. The species is present in most of South Florida, and its northward expansion has been evaluated via eDNA. Over the years, USGS has spent a lot of time studying this snake with its partners, and we've learned a lot. And here are some of the attributes. This is a large body constrictor, easily able to get up to lengths of 20 feet. It's a generalist predator. We're going to cover more of that in a few minutes. It's highly fecund. It has sexual maturity at an early age. It seems to be extremely adaptable. It has high tolerance for extreme environmental conditions such as salinity, heat, and it can withstand cold to a certain extent. It has regenerative organ growth, so the digestive GI tract shrinks back until it has a meal, and then those organs grow. It has multiple paternity. And while we have learned a lot about its basic biology, we have major gaps in our knowledge, and knowing their vital rates is one of those gaps. And that's going to be an important piece for us to better help managers manage this species. So our current research priority is obtaining python vital rates. And we're looking at age and sex and size specific survival rates, sex ratios, the age and size at maturity, the reproductive output and frequency. We're looking at population growth rates, how they just from the nest site. And all of this research is also informing a synthetic biocontrol aspect to this work that's led by Maggie Hunter and Adam Perez. And inadvertently, because we have eyes and ears on the ground, we're able to begin to assess the impacts of this invasive snake at deeper and deeper levels. So one of the things that we can look at when we are on the ground for the vital rate work, where we have snakes that are radio tagged, is when we follow these snakes, we can see when they've eaten a meal. And here pictured at the white arrow, it's pointing to a food bolus. This python has recently ingested a prey item. And to the right is kind of our field notes of going back and checking on that bolus. And when that food bolus gets lower down in the GI tract, we can actually palpate the snake and have it express the food bolus. We can collect it and then dissect it. And we were able to confirm that food bolus on October 16th of 2023 was in fact a deer. So Kristen is going to finish our presentation with you by sharing information on the biodiversity impacts and the additional research avenues that we have going with our collaborative to get at this issue. So over to you, Kristen.
Okay. The impacts of pythons on native wildlife is truly a major concern for all managers. The pythons do directly compete with natives for food. They actually have a similar diet to the Florida panther in many ways. They are top predators, and they're consuming other top predators such as alligators. They do consume many mammals and birds, and we'll show that in a slide in a few seconds, but they are consuming many of the key iconic Everglades species that we are finding in their diet. In the 2012 Proceedings of the National Academy of Sciences paper, we showed that over 98% declines in available data for Everglades National Park raccoons and opossums were occurring, and we had severe impacts also to marsh rabbits. Our other colleagues focused on studies on birds, and they showed predation on wading birds as well. And these images are from both a camera trap, one of the wading bird photos, and then on the right is what we're most concerned with in some of our locations, which is impacts to deer populations. To illustrate the impact of their diet, a colleague from the South Florida Water Management District put the figure on the right together, and it's essentially, what does it take to get to a 13-foot python? The left-hand side is about a 13-foot python with a bunch of our key interns and employees, just to get a sense. Peter on the right is a 6'6" individual. And so you can see the impact for any one snake is pretty large. On the biodiversity theme, we have several varied studies on impacts, as Amy mentioned. We do perform necropsies to identify specific diet items, which we can do through remaining scutes, hair, and feathers after the digestive process. We are also doing stable isotope analyses because we have a tail retained from every removed python. We can extract tissue from that. And we're looking at the ecological role of the pythons across that range or their niche. We're spearheading the use of accelerometry, which is a very fine scale activity and behavior sensor. We put these in pythons in the wild, and we're examining things like feeding frequency. We are, as I mentioned, we have field cameras deployed in certain areas, and in some cases, we're detecting feeding events. And we are paying particular attention to any food boluses, as Amy pointed out, that are observed during our normal radio tracking field work. These photos on the left show an American coot. In the middle, it's a white ibis, and on the right is a little marsh rabbit. The photos on the right are actually from field cameras deployed in the Florida Keys, where there's a tiny little mouse in there. And then the python is coming out of the pipe on the right, and it consumes it. And this is occurring at a pretty higher rate than we expected. Essentially, almost any native endotherm in South Florida is vulnerable to python predation. So we have quite a biodiverse diet. This is a reproduction of a figure from our 2023 Python synthesis led by Jackie Guzzi, and the data releases from Christina Romagosa, our near and dear UF partner, and it shows the diet data collected for this damaging species. So across 2,100 samples from 1,690 pythons, we saw 71 prey taxa, 25 mammal species, 44 bird species, and 2 reptile species. These are data from 15 years of necropsies, so this is a lot of work. The numbers along the x-axis are the number of prey found within the digestive tract of pythons. And I should mention that digestion is very acidic. So in some cases, We can't really enumerate what they ate, just that something was present. Not much remains after the highly acidic digestive process. And then we do have ones that are also empty. So far, we've published one paper on sort of an exciting aspect of our trials with accelerometers, and we're now using what we've learned there to obtain more what I call validation observations to interpret the data that's logged in animals in the wild. The colors in the figure on the left show hours moving, with warmer colors indicating more, and then the gray box actually on that first line is when this particular python died. So this is the type of information we can get from these devices. The right photos show the accelerometer on top and then the placement of it on the bottom with like that small hand. They're about the size of a USB stick, which is exciting. And we do implant them right under the skin. Our observations are key really to understanding feeding events because we don't see them that often in the wild. So we have the potential to interpret interesting behaviors that we see in the data. Today, in fact, we did 2 surgeries on pythons that are part of Amy and my study. And these are in areas with deer as prey. So our hope is that we actually capture any feeding events on these tags that we can pair then with direct observations of these study snakes that may have food boluses in the near future. This is what we're doing right now with a student, Isaac Lord, and some other collaborators. We're actually doing field validation trials. We're feeding pythons that are previously wild pythons, but they will eat. We're feeding them rats, and we have the devices, we have these accelerometers on or in them. And so we can see when the animal strikes, and we have the date and time stamp on that, and then what's called pterygoid walking as the animal further envelops the prey item and walks up the prey. The next slide will show one more observation of this. So we can actually determine a defensive strike based on our observations versus a feeding strike. And this is just an example of what that data looks like. Many partners, as I mentioned and Amy mentioned, are involved in this work, and we wanted to acknowledge each and every one of them. And then we'll wrap up with 2 more slides. We are really making progress towards informed management on the python front. Our collaborative vital rates project is allowing us to collect key data from 2 sites in particular, and our multi-year study with a now diverse funding portfolio is really needed for this work. It may still only be a shoestring compared to other lines of funding for damaging invaders. We are excited that we're getting the needed baseline data for this species in order really to improve the clarity in management decisions, particularly the pieces of that python life table. So together with managers, we can better interpret removals. We are quantifying the impacts to biodiversity across the landscape in multiple ways. And of course, as has been mentioned already, there's no silver bullet with this species either. We thank all of these individuals for funding. And I want to thank Mark Ian Cook for his incredible photos of the landscape. And I think we might have time for a few questions.
Kristen, that's incredible. Oh my gosh. Thank you so much for being here and sharing all this work. I'll just say, I think it's probably obvious. Most everybody who listens understands this, but it's worth saying. Everything you see here, everything that we do is done with really large teams and in conjunction with on the ground folks. So none of this is done in a laboratory by a scientist alone. It's all done in partnerships. Thank you so much. A couple of questions. One, since pythons are such an important issue, is there anything being done about exotic pet trade? And is there anything that can be done to limit that?
I believe it is quite limited now. It wasn't for many, many years, but now even interstate trade, the permitting process is onerous. And so there are not nearly as many as there were, even 12 years ago coming into this country or being traded.
Yeah.
Okay. Another question, what is the synthetic biocontrol form for these python?
I think Amy and I both can answer. I don't know if Maggie's on, but imagine a way where we could either shut down reproduction or cause them to have heart defects. It's basically something we can do to affect their biology where we kind of break it down. And so Maggie is exploring that. This is a common thing with invasive fish in other countries where you can basically introduce animals that will only have male offspring. I'm not sure that's the exact path we're headed, but if there's a way we can shut down their success, either in feeding or breeding, I believe that's the direction. Amy, maybe you want to comment as well.
Yeah, I would just add to that, Kristen, that, this team regularly interfaces with Maggie and Adam so that we make sure that we're getting the information they will need to inform the synthetic biocontrol of this species. So it's a constant dialogue.
Yeah. Well, I want to just say, you would think we have survival rates and reproduction rates. We actually don't have that for this species. We're figuring that out now.
Yeah.
So even though this is such a damaging species, we don't know a lot about, like, what we know about their biology in South Florida, we're using but we still are in the baseline, figuring out some of this. How often do they breed?
Mm-hmm. Yeah,
that
is definitely an unknown. Yeah.
I'll just in closing for this talk, I'll just mention that this work is being done by at least 2 USGS centers, as well as lots of folks in Florida and others nationally. So it's a huge team, and you guys are just doing such great and important work, and I really appreciate it.
Thank you for your support, and it was fun to present some of it.
It's always great to hear from y'all. There may be some more questions in the chat you can answer. Thank you so much. Our final talk before our roundtable is from Kurt Kowalski from the Great Lakes Science Center. And Kurt's going to be talking about common reed control.
Thanks, James. Appreciate it. I'm happy to anchor this great set of presentations here by USGS, just really highlighting, as James said, the collaborative work that we do. You're going to hear the same message from me as I get to talk about the work we're doing on the invasive common reed that is a scourge of the Great Lakes, and it's really all over North America. So I will say that I get to be up here to talk to you, to talk about this collaborative work that we're doing that is really being driven by the Great Lakes Restoration Initiative, funding from GLRI, but then also USGS and other partners that really make the work, the other names you see on the slide, make it all work. So it's very collaborative, and a lot of team work has gone into what I get to show to you here today. So I get to talk about Phragmites australis, specifically subspecies australis. It's a perennial grass. So unlike the grasses that Seth was talking about earlier, this is very tall, very robust, a lot of above and below ground biomass, thick leaves, big seed head. There is a native to North America, so we do need to be a little careful as we talk about this plant and identify it in the field. But for my talk and most everything, when I just talk about Phragmites, it's about the native subspecies. This subspecies is, like many invasive species, causes big impacts to ecosystems and people, and dramatically reduces biodiversity in many ways. As you can see from the pictures there, it grows very densely and can crowd out native plants, make it difficult for wildlife to get through, but also impacts people. That image on that upper right-hand corner there, that's a 2-story home. That's Phragmites growing on a beach in the Great Lakes. So impacting recreation, it's a fire hazard, and it has a lot of impacts on people and the biology that we study. So here's an EDMAPS, a map of distribution in the green there, that just to recognize that this is not just a Great Lakes issue. I'm speaking from the Great Lakes at the Great Lakes Science Center based out of Ann Arbor, but working on an issue that really impacts all of the US. The reality is this is a North America problem. And it is among the top priorities for a number of different agencies. Again, the GLRI, Great Lakes Restoration Initiative action plans, invasive species and Phragmites are called out, various other plans at different levels, provincial, federal, provincial, state, and further on down the line. So this is a high priority, and in many ways, it competes in ways like the cheatgrass and kudzu and many other plants that people are familiar with. Because of all that, and GLRI initiating around 2010, people in the Great Lakes Basin recognize the need to work together, share information about this particular species. And so we at the USGS work with the Great Lakes Commission to help stand up the Great Lakes Phragmites Collaborative to really organize the efforts that were going on, not just research, but management and communication, policy, and so on. And we developed this website, greatlakesphragmites.net, that you see there. I encourage you, even if you're not in the Great Lakes, go check it out. There's just a tremendous amount of resources about this plant and how people are managing it, case studies, best management practices, all that type of thing. And so the collaborative is way more than the website, but the website really represents a lot of the products that the collaborative has been identifying the need to spend energy on. So one of the priorities that the collaborative has pulled out is thinking about management, how to control this invasive species given what we know about it. There are conventional approaches that work pretty well, but there's uncertainties about what's the best combination of these treatments in order to get the desired result, recognizing that there are differences in site conditions, the ways people implement them, the rules and regulations around implementation. And again, there's uncertainty about which one of them work best on the landscape. So that's where USGS, my team, and others worked with the Great Lakes Commission, worked with the University of Georgia and others to help stand up what I end up calling the Phragmites Adaptive Management Framework, really targeting on reducing that uncertainty around which management approaches work best given site conditions. And as the top graphic kind of shows, we're learning from all those individual actions on the landscape. That is informing a model that then provides guidance back to people on the landscape to then help them make decisions about management. And then the cycle continues to get better and better information driven by actual responses to treatments. And so that there's also a whole lot more I could talk about there. So these programs are very useful to look at these conventional approaches, but the theme that came out over and over again through the years was they really just need more tools in the toolbox. And so I'm going to talk about three of them here. The first one is here just in one slide because I don't work on this, but I want to recognize it as we think about innovative approaches for invasive Phragmites. And the first is the classic insect biocontrol that there are folks in the States and Canada that are working on developing insect-based controls. And the only other thing I'm going to say is there was just a talk about this that Michael McTavish talked about this at the Ontario Phragmites Working Group meeting just a month ago. So you might go check that out for kind of the latest and greatest. But I just want to recognize that as I get to talk about the research that I get to be involved in. And that's what I'm talking to you here about for the next 15 minutes or 10 minutes, whatever, to talk about the role of the microbiome and symbiotic relationships between microbes and the plants and genetic biocontrol targeting natural defense mechanism in the plant. So let me first talk about the microbiome here. So, as time goes on and technology gets better and better, we learn more and more about the importance of microbes, bacteria, fungi, and others in human health. And we can really learn a lesson from that because we recognize now more and more microbes are in us and around us and on our skin, and that balance is needed in order to maintain human health and many bodily functions. And so, again, bacteria and microbes occur all throughout the human body, just as they occur all throughout plants. So in the stem, inside the cells, in the roots. And so we're really evaluating those microbes and the benefits they confer and targeting those as a form of control. So I just offer a paper here that's in Science. So this is pretty well documented, this symbiotic benefits that plants and microbes can help. So the target of our work has been really, can we disturb microbes in order to control Phragmites? So in 2012 or so, maybe a little bit before, we really started down this formal collaborative approach to maximize the collective impact of the research that's being done on microbes, microbial symbiosis. We created a group that really focused on these microbiome research gaps. Like, what do we not know? What do we know? What do we not know? And then how can we move that towards a microbe-based control or treatment for this invasive plant? It's been very collaborative. There's been a lot of science done on it, a lot of papers that have really just been focused on inventories and these fundamental blocks to work us towards a microbe-based control for this plant. And it's produced some really cool things, including a patent, another patent application for a bioherbicide that I'm going to talk about shortly. So this is, again, a very collaborative approach that represents the type of science that we do for this very real problem. quickly highlight this paper. Again, if you're interested about this approach and this collective impact approach, this is a paper we did in 2015 that with all the various partners that really lays out the research agenda that the broader team is working on. And one of the big outcomes from that effort and the collaborative effort is working towards development of a non-toxic bioherbicide that we've been working on primarily with Rutgers University, Dr. Jim White. And this is focused on disrupting the microbial relationship within the plant. And so all of the components are all things that we use in human health or are non-toxic, citric oil, glycerol, alcohol used as more of a surfactant, arginine, which is an amino acid, and then sucrose, sugar, and then microbes. And so then there's this mixture that we're applying to the plants in an experimental way to really evaluate the impact and evaluate how well this might work as a new tool, as a new approach to add to the mix with the other treatment approaches that are out there. So here's our approach over the last number of years of developing this bioherbicide, applying it in the field to stimulate microbial response and disrupt the natural balance that's there, monitor the plant response, and then look at it, refine, and do it again and improve things through time. The impacts are pretty clear and pretty visible. Within 1 week, we're seeing browning of the tissues, the above ground biomass tissues. You can hopefully see the brown leaves there, there's curling and visible impact. And then throughout the growing season, we see this at the plot level. These are 1 meter plots with the control on the left, so only water there. And then very frequent, every 2 weeks using the treatment. In the middle pane, you see a lot of brown. Phragmite, this is taken at the end of the growing season, and then every 4 weeks on the right. So you can see visibly that, and we have the data, the quantitative data behind it to show that it's having a pretty big impact to the above ground biomass. And so we're continuing to evaluate the data, of course, that we're collecting to improve the mixture and the application approach. We're seeing that there seems to be a pretty good contact effect, but we're really evaluating the systemic nature of it and looking to continue to tweak and modify the formulation and the application approaches through time, and really expanding partnerships to develop this from this experimental stage into something bigger and make it more available to folks. So that's really exciting work that's been going on. A lot more could be said about it, but I want to share it with you as one optimistic or potential positive option that's coming down the road. So now let me just here finish up by talking about the work we're doing, what we're calling genetic biocontrol. So if we think about Phragmites, and we can recognize that a lot of the traits that Phragmites has are what make it so invasive. It's super tall, has these very, very dense stems, incredible amount of biomass below ground with roots and rhizomes. It has the seed head you can see. And so these are the traits that help make it so aggressive and outcompete native plants and impact biodiversity and such. So the target of this work are those traits and expression of those particular traits. And the question is, can we switch off expression of those traits and make that as a form of control? This work is really founded around some genomic work that we did. So genomics being, studying of the whole genome. So all of the traits together, all of the genes and the whole genome together. We were able to put out a paper with the LSU team, Louisiana State University team, looking at and describing the genome for the invasive Phragmites. And we're almost done with the work with the native Phragmites genome, and then being able to compare those will really allow us to target very specific parts of the code and to make it as species specific as possible, but also to really understand what characteristics of the invasive subspecies make it so competitive compared to the native. So we're working on that aspect of understanding the roadmap, if you will, of the traits and genes. And then we're applying technology that's been quite well described and established called gene silencing. So gene silencing is focused on the RNA of the plant. So not the DNA, it's the RNA of the plant, which I'll talk about a little bit more, which means that it's transient. There's nothing to pass on to the next generation. We're just talking about blocking expression of certain traits of the DNA that code for these critical plant processes that help it be competitive: photosynthesis, flower development, root development. And then if we can limit the expression of those traits, then the plant should be less dominant, and some of the other plants should be able to compete with it, or we may be able to cause the plant not to be able to reproduce or be able to photosynthesize or have these other critical processes to live. So now I'm just going to show a quick animation here at the super high level. Some of you may have seen this, but I just think it's pretty effective to describe how this technology and this approach is different than others that we've heard about earlier today, but also are out there being discussed and worked on. So we know that DNA is this foundation, and that's the roadmap that the information gets transcribed into messenger RNA, these single-stranded RNAs that get translated to proteins, and then you get the traits. It's the basic process. So if you don't want those traits, we can kind of work backwards here and target those single-stranded messenger RNAs. So we've been following this path and adapting it for invasive Phragmites and ultimately for other invasive plants is the idea. Because it's we're adapting this as it was first described in this Napoli et al. Paper in 1990 that really laid out these purple petunias that they were working on, and they said, hey, if we could make them more purple, then we will be able to make them more productive and be more desirable. So they added the double-stranded RNA to the plant and found out that they turned white. Very confusing. So dug into a whole lot more and ultimately recognized that there's this natural plant defense mechanism that when a plant detects double-stranded RNA, it will degrade alongside those complementary single strands. And this was later formally called RNA interference and received a Nobel Prize in 2006. So if we go back to this little graphic, if we were to hypothetically introduce a photosynthesized double-stranded RNA, the plant wouldn't like it. It would come in and disrupt that whole process, ultimately getting a plant that doesn't photosynthesize. So that's the theory behind what we're doing, and implementing it through time ultimately gets us to this species-specific and non-heritable or nothing to pass on to the next generation type of approach. So let me finish up here just by saying that we're applying it to invasive species, but it's already being used in human health. It's being used in agriculture. It's being used in other pest developments for other insect pests and so on. And so this work is really going on with the Corps of Engineers, really being led by Dr. Ping Gong and his team at ERDC, Engineer Research Development Center, Vicksburg, Mississippi, as we work to develop these nanotechnologies that get these RNA-induced gene silencing agents into the plant that then produces the silencing that we're looking for. Very encouraging results so far. Green is normal, blue is treatment. You can see we are seeing that impact of this effort in the plant signal. And then in the bottom right, you see the curling leaves. We're also seeing it phenotypically. So just continuing to work out that process of what are the optimal components, applications, and genetic targets that we're really looking for to develop this into a really usable technique and control for the invasive Phragmites, and then use that to pivot as a foundation for other invasive plants moving forward. So my contact information is there. I'll put it in the chat. James is up, so I will take questions at this point. Thank you.
What incredible work. You guys are on the cutting edge of science. You are inventing science, and I really, really appreciate the work you're doing. It's sexy. It's awesome. It's helpful. I have one question, and the rest you'll have to answer in the chat there. Is this a one and done type of treatment that you're talking about, or is this something you'd have to repeat every couple of weeks, or a couple of times a season, or every year? What are you thinking on that?
We are testing this experimentally right now at a very high frequency rate to show proof of concept and to really evaluate the impacts of the treatment on the plants, recognizing that the rate and approaches that we're taking right now aren't likely to be feasible at a large scale and economically feasible moving down the road. So the idea is to test out the technologies now and then work towards that adaptation to a more available and usable product.
Thanks, Kurt. Thank you all for being here. This is the end of my session, and we're going to move over to Patrick Kowalski and Paul Heimowitz, my colleagues in the Biological Threats and Invasive Species program at USGS. And I'll be signing off, turning it over to them. Thank you for your engagement. It's been great.
Thanks, Kurt. Thanks, everyone, for your presentations today. Going back to the theme for this year, which was invasive species and biodiversity, we wanted to have our panelists, our speakers, try to talk a little bit more about that. Each of you did a great job of tying in the biodiversity issue in invasive species. But we'd like to have a little more discussion on that for our audience. And I'm going to start with a question that Paul and James and I have been working on here to help kick things off. So we'll premise it with the relationship between non-native species and biodiversity can seem counterintuitive or almost paradoxical at first. You add a new species to a system, you've increased your biodiversity. And because not all non-native species become invasive, that creates, that could create in some minds the idea that, so what's the problem? So for each of you, each of the panelists, thinking of the species and ecosystems you study, and given that global change is affecting native biodiversity, how do we manage the risk of new introductions? So that's a general question. Kurt, you're up on screen now. If you'd like to start, that's great. Other panelists, feel free to either activate or not activate your cameras and join the conversation.
Yeah, managing new introductions is tough, and there's a lot of energy that's going into that already. And even in the Great Lakes region where we're at, there's a lot of shipping traffic, there's a lot of movement of commerce, there's a lot of people that move around, we have lakes that move seeds, we have all kinds of opportunities to receive new species into new territories. And there's already a lot of work going on at the federal, state, at all levels of management and home ownership or land ownership to identify and then implement to the best possible approaches to minimize spread and that true invasiveness that really impacts the ecosystem at a large scale. So that's not what I work on, but I'm fully aware that that's key to ensure that treatments that like we're working on aren't needed at a large scale, but might be very useful at those very small initial populations.
Thank you, Kurt. Diane or Kristen or Amy, any of you have any additional points to make on the subject of managing new introductions?
Yeah, I would just quickly add the importance that I've noticed in some of the work that I've been doing, not with the python, but with the invasive tegu lizard, and taking sightings that are appearing throughout the Southeast. And we've recently modeled with climate projection scenarios that while you may not have an established population under a 2-degree warming, you certainly could. And reaching out to the state agencies and having that conversation, letting them know about the work has been really an interesting way to go about preventing. And so this has been something that I've been doing for a couple of years now. And states in the South have gone ahead and banned tegu lizards before they're sitting on established populations. So I'm trying to work at that front end before they get established.
Mm-hmm. Yeah, that's a great point, Amy. Once something's here and wreaking havoc like Burmese pythons, that's a much different challenge. If you can stop them in the first place, you're so far ahead.
I was going to add to that. I recently reviewed a paper on an invasive turtle species that were detected by AI. So I do think there's a real opportunity at our ports to improve detection in a scientific way. Mm-hmm. But we also have to understand that our lists are imperfect with what we think is here and what's going to make it here, because we are surprised sometimes that something is even in this country and it wasn't on the list, and it might have, a similar type of reproduction or diet or preference as the Burmese python. And so I think we have to be looking at the suite of traits and the suite of possible environmental characteristics in which these species can make it, that might be a little more flexible than just no or yes. You know, it's quite a bit that these animals can do behaviorally as well to make it here, at least where I sit. I'm in South Florida.
An invasion hotspot.
Mm-hmm.
Right. And of course, Kristen speaking indirectly about risk assessments and about horizon scans, which is something James can speak about with some degree of expertise. That's part of ongoing work that USGS has been doing for a long time and in collaboration with numerous partners. Clearly important to not only prevent things from coming in, but know what you need to be looking for to prevent coming in. So Renee and Seth, any observations from your parts of the world, islands and western arid regions, southwestern arid regions?
Yeah, if I could hop in with a comment. Kristen, you brought up the cargo, like shipping cargo. And so Hawaii, of course, is at a port and there's a lot of transportation around the Pacific. There's a strategic relocation of troops to Guam. And so there is just a lot of potential for different organisms to get transferred around. So some of the work that we're conducting is through the Biothreats Program and also a bill-funded project, which is developing techniques to interdict invasive species from shipping cargo, and then develop tools for assessing whether or not an invasive species is hitchhiking within a given cargo container or, you know, some kind of a ship transportation process. With the tools like point of use assays and CRISPR, there will be more opportunity for developing tools. However, as Kristen also pointed out, those are often species specific. And so for a point of use detection tool, it would be quite targeted. But, you know, we're just kind of at the beginnings of this type of research. And so hopefully in the future, we can have something that's more general.
Yeah,
Thank
Patrick,
you.
I'll just add that, you know, we're not at a port of entry in much of this Intermountain West, but besides the, you know, newer technologies of detection, as far as invasive grasses go, some of it can be fairly simple. There's some new effort to just look at the effectiveness of boot washing stations, because oftentimes we walk through area with invasives and then enter an area that's not invaded, or potentially doing the same with tires, because we know that most invasive grasses start from roadsides. Their spread is a lot of times initially limited to those areas, and that's potentially where we have the greatest potential to either eradicate them or at least control their spread. So in addition to kind of some of the more advanced ways of detecting and dealing with them, I think some simple options like that could be helpful as well.
Yeah, thank you, Seth. And I'll just point out that you may not have ports of entry, but when one just looks at a map of national park and national monument distribution across the United States, the vast majority are west of the 100th, which is where the invasive grasses are a big deal. And there are millions of people coming into the Western United States from all over the place. They come with their boats, they come with their kayaks, they come with their trailers and things. And these are vectors for movement of invasive species as well. So it may not be a port of entry, but there are certainly vectors for entry. Okay, thank you. Any other points to be made by the panelists?
So thank you for the opportunity for being able to present today. It's a pleasure to be able to reach this broad of an audience and to describe the work that we're conducting. So thanks.
Yes, agreed. Thank you, Renee. It's great to be able to have this partnership, annual partnership with NISAW for National Invasive Species Awareness Week. So at this point, we're getting close to the top of the hour. So I'm going to kick it back to my colleague, Paul Heimowitz, who will have a few closing remarks for you. And then after Paul's finished, we'll have a final goodbye from our gracious host and coordinator, Elizabeth Brown. So Paul, if you're available.
Thanks, Patrick. So Paul Heimowitz, I'm the Terrestrial Invasive Species Program Manager in the same biological threats and invasive species group with James and Patrick. We sometimes think of ourselves kind of as the three musketeers. That's true with today. And I will, on behalf of all three of us, just extend huge thanks. I hope that these presentations have wowed you as much as they have me. And it's just really a great honor to be associated with all of our amazing invasive species scientists here at USGS, but particular thanks to the fabulous presenters who you heard from today. And of course, a big thanks to NISAW, to Christine, Elizabeth, and others who have helped host this annual USGS webinar during National Invasive Species Awareness Week. We look forward to this continued annual tradition. And more than anything, so much gratitude to all of you who have joined today. We had a great turnout, and we appreciate that this is being recorded, and others will eventually get a chance to hear this great information as well. And we're always interested in hearing from our partners and all of you in the audience today about new opportunities to increase awareness of our invasive species science. Please feel free to reach out to us if you have ideas or questions. As you've heard many times today, biological invasions, are such a huge challenge, but when we get this great collaborative scientific research married with the broad partnerships of natural resource managers and educators and just all of us in our role as community members, can really lead to some major successes in prevention, detection, and control that protect native biodiversity. And so with that, I'll hand it over to Elizabeth with NISAW to close us out.
All right. Well, thank you so very much. Thank you to James and Patrick and Paul and all of our speakers today. You so clearly articulated the importance of invasive species prevention and management for the protection of our biodiversity in North America. So it was just an outstanding start to National Invasive Species Awareness Week 2024. Thank you for being here today. We have daily webinars each day this week, and I hope that you will take advantage and join us for more events later in the week. Tomorrow is the Invasive Species Language Workshop, which is being sponsored and hosted by Sea Grant in Washington, D.C. We have the great privilege of being able to broadcast that event to those that are not able to travel. I hope that you will join us for that important conversation tomorrow. On Wednesday, I'm very pleased to be back with Paul and some of our friends here hosting FICMANU, or the Federal Interagency Committee on the Management of Noxious and Exotic Weeds. So you'll hear from numerous federal partners on their important work in the noxious weed space. Thursday, we have the opportunity to hear from experts on opportunities and challenges for preventing the next plant invasion. That webinar is being sponsored by CAST, and we're so excited to hear about a new paper that's coming out there. And then finally, on Friday, you'll get to hear from some of our NISAW members and experts. We've had a subcommittee working on a new position paper on biodiversity, and we'll be sharing that with you on Friday. So please join us every day this week. Go to NISAW.org. Our events calendar has not just our events, but events happening all across the continent, including local events in your area. So please check that out. Take advantage of the NISAW toolkit and take action at one of our Take Action links. And so with that, I will conclude by expressing deep gratitude to our NISAW sponsors, without whom this week would not be possible. On behalf of everyone at NISAW, we sincerely appreciate your support. And that concludes our 4th annual USGS NISAW Invasive Species Research Forum. Thank you to our speakers, our participants, and our organizers. We appreciate the work that you do every day and are grateful that you're a part of the invasive species community. So thank you, everyone. Happy National Invasive Species Awareness Week, and we hope to see you again tomorrow or one day this week.